So today we're going to be covering transcription and translation. This is chapter 10, modules 10.6 to 10.16 in your book. So this is just a reminder from last lecture. So remember we studied the structure of DNA and RNA. So deoxyribonucleic acid is DNA and ribonucleic acids are RNA. Specifically, we studied the structure, directionality, and the major differences between DNA and RNA. In the second part of the lecture, we focused specifically on DNA replication, and we learned some of the major players in that process. So we learned about enzymes like DNA polymerase, helicase, ligase, and a few more. Today, however, we're going to be focusing on transcription and translation. And as you're soon going to see, those two processes are very important in taking us from DNA to RNA to protein. So to get you guys primed for today's lecture, here's a question for you to answer on Blackboard. So which of the following is a function of proteins? So this is just a reminder that proteins have many functions. So they need to do all the work that needs to get done in the cell. So as you can imagine, proteins being such important molecules for the cell also makes the process of making proteins themselves very important. And that's absolutely true. So we're going to be spending a whole entire lecture today figuring out how exactly we make proteins. And again, that's going to be through the process of transcription and translation. So when I give this lecture in person, I usually like to talk about an analogy I use. And here the analogy relies on this situation here, where you're trying to build a car from scratch. The reason why I like this analogy is because it actually explains a lot of the reasoning behind why the cell does transcription and translation. So I think some of the processes, as you're going to see, are kind of counterintuitive. So why does the cell go through all this trouble? But hopefully once you walk through this analogy, it's going to make more sense to you. Okay, so let's pretend you want to build a car from scratch. We're also going to pretend that this is the 90s or 80s or before, when you didn't have access to the internet to do everything. So naturally, most people will visit a library. So that's where people got their information knowledge long before the internet. And this is a good place to start because libraries contain lots of information on different topics. So you need to learn about engineering, you need to learn about electrical work, about steel work, and the library makes a good place to start because it contains lots of information about many different topics. So if you're building a car, you need to be able to do more than just weld steel. You need to be able to do electrical work. You need to figure out how to do mechanics, how to build an engine. So it's much more complex than just a single topic. So you need somewhere where you can go to find all this information you need. And the best place for that would probably be a library. So I said you need to learn a lot about different topics. And probably a good place to start would actually be the encyclopedia. Again, you'll need to study a lot of different topics. So things like M for motors, S for steel, so on and so on. And the encyclopedia can be a very helpful resource for that. So this is an issue though. So you want to use the encyclopedia and you're absolutely able to do that inside the library itself. However, as you may know, it's not possible to check out reference books like the encyclopedia or a dictionary or a thesaurus or anything like that. So the encyclopedia has to stay there. And I think the reason why libraries do this is because they don't want any pieces of the encyclopedia to go missing or become damaged. So an encyclopedia set is actually very expensive. So the library wants to make sure that's protected, and they don't let people check that out because of that reason. So then what would you do in this case? So you can't check out the encyclopedia, but you want to use it, and you want to bring it home so you can use it specifically in your garage to build your car. It's not much use to have an encyclopedia in the library, and you have to go back and forth to the library to find the information you need. So it'd be very nice if you could bring that information to somewhere useful. So as you may know, most libraries have some sort of photocopier around, and that's very convenient. So you can actually make photocopies of sections of the book you want to use. So that'll be very helpful for trying to get the encyclopedia outside of the library. So you can actually make copies of the sections of the encyclopedia you want to take home. And a follow-up to that question is, would you want to copy everything from the encyclopedia? So do you want to copy the whole entire encyclopedia set? or would you just copy parts of it? And of course, the natural answer here is you only want to copy parts of it, especially if you're using a library photocopier, which probably costs about 10 cents per page. You only want to copy the sections you absolutely need. If you're going to copy the whole entire encyclopedia set itself, you might as well just buy your own set. Okay, so now you've made your copies of the sections you want. You can now take those papers home and start reading. But as you can imagine, looking at an encyclopedia with information in it, it doesn't tell you step 1, step 2, step 3, this is what you need to do to build a car. So you have to translate that information to something useful. So that is, you have to take the ideas and information found in the encyclopedia and turn that into steps that you can actually use to build the car. But once you've done all that, you'll have your car built and you can start driving it. So as I alluded to at the beginning of this analogy, all the steps I've just discussed are heavily related to the processes of transcription and translation. So the library itself is much like the nucleus of the cell. So as you know, the nucleus contains the DNA or information for building things. In the case of the library, for building a car. In the case of the nucleus, for building protein. However, the DNA cannot leave the nucleus, just like the librarian doesn't want the encyclopedias to leave the library so that they don't get damaged. And because of that, we have to go through the process of transcription. So transcription is just like making photocopies of the encyclopedia. So transcription takes a small chunk of DNA and creates RNA off that DNA. Just like we took a small chunk of the encyclopedia to make photocopies of, here we're taking a small piece of DNA and making a photocopy of that. The benefit of this, though, where your photocopies are able to leave the library or nucleus. In other words, the RNA is able to leave the nucleus and enter the cytoplasm. So the cytoplasm is like your home. So it's actually going to be the place where you're going to be doing the process of building your car. But as we said, though, the encyclopedia doesn't give you a step-by-step directions on how to build the car. So you have to translate that information into something useful. And that's exactly what the cell does. So the RNA contains information, but the cell has to translate that RNA into amino acids or protein. This is similar to how you have to translate the information in the encyclopedia into useful steps for building a car. And once you have your car, you have your finished protein, and it's able to do work. In this case, the car is able to drive, and your protein is able to do one of the many functions that proteins can do, some of which we discussed in the first question of this lecture. So hopefully that analogy is very helpful for explaining the processes of transcription and translation, and some of the rationale as to why the cell actually goes through these processes. So here's the big picture. So essentially what we've just gone over. So we start off with DNA in the nucleus. Remember, the DNA can't leave, so it stays there. However, through the process of transcription, we create RNA based off that DNA. Transcription happens in the nucleus, but once transcription is done, the RNA itself can actually leave the nucleus and enter the cytoplasm of the cell. In the cytoplasm, that RNA is translated into a protein. So again, we go from DNA to RNA to protein. DNA to RNA is called transcription. RNA to protein is called translation. And here's exactly that, just written out in text form. So transcription is the synthesis of RNA under the direction of DNA. So essentially DNA to RNA. And translation is the synthesis of proteins under the direction of RNA. So essentially RNA to protein. So here's a little bit of terminology. So we already discussed a little bit about what genes are in earlier chapters of the book. But here's an actual specific definition for you guys to remember. So genes provide the instructions for making specific proteins. So that's the definition I want you to remember. If you take a different biology course or use a different textbook, you might get a slightly different definition of what a gene is. But for this course, we'll use this definition for a gene, so they provide instructions for making specific proteins. So next we're going to talk about codons. So this is something we haven't talked about yet. So codons are the basis from going from RNA to protein. So remember, we go from RNA to protein through the process of translation. So codons are how we translate RNA into amino acids, which make up the protein. And codons themselves are based on a triplet code. So remember, RNA is made up of nucleotides. So each 3 nucleotides is a triplet, and we call each of these triplets a codon. These codons then specify specific amino acids. For example, if we had the codon AUG, so adenine uracil guanine, that codes for the amino acid methionine. So each triplet code of RNA, or each codon, specifies a certain amino acid. And as we're soon going to see, some amino acids are specified by multiple codons. So for something like lysine, that has 3 or 4 different codons that specify lysine. So we'll learn more about codons a little bit later in this lecture, but here just be aware they're triplet chunks of RNA. So really the reason why I decided to define those 2 words is for this figure right here. So here we have some DNA, which remember is the heredity molecule of the cell. This DNA itself has multiple genes on it, so gene 1, gene 2, and gene 3. And each of those genes, remember, codes for a protein. So here we're zooming in specifically on gene 3. We can see it has a DNA sequence. Remember, DNA is normally double-stranded, so we'd actually have a complementary strand, but here we're only zooming in on one of the strands, since that box is not around the bottom strand. So based on this DNA, we're going to have the process of transcription happen. So as you can see, we go from DNA to RNA, and if you look closely, we actually have complementary base pairing happening. So here we have A pairing with U, G's with C's, and so on and so on. So remember in RNA, instead of thymine, we have uracil. So here we have AU pairing and GC pairing. Also, we talked about codons, which you can see here. So remember, codons are triplet pieces of RNA. So every 3 nucleotides, we have 1 codon. So you can see UUU is 1 codon, and that codes for the square amino acid. GGC is another codon, and that codes for the circle amino acid. Also, just a quick reminder, a polypeptide is a bunch of amino acids linked together. So remember, poly means many, and peptide relates to the bond that links amino acids. So a polypeptide is many amino acids linked together. When that polypeptide folds into a 3D shape, we then start calling it a protein. But here, for the purposes of this class, polypeptide and protein can be used interchangeably. So here's a very similar slide to the last one I just showed you. So here we actually see both strands of DNA. It's very important to note that RNA can be made from either the top strand or the bottom strand. So it's not always the top strand, it's not always the bottom strand. It depends on what's called the promoter. So the promoter we'll talk about later in this lecture. But for now, know that I can use either the top strand or the bottom strand, depending on the location of the promoter. Here, though, we're using the top strand. So again, we have transcription, and you can see there's base pairing between the top strand and our RNA. So A is pairing to T, U is pairing to A, G to C, so on and so on. So since the top strand is being transcribed, we normally call the top strand the template. So here the top strand is working the template, just like we saw both strands of DNA working as a template in DNA replication. So similar idea and similar terminology here. The bottom strand, though, since it's not being transcribed here, is sometimes called the non-template strand. So a pretty intuitive name there. Okay, so this figure actually goes in a little bit more detail about the process of translation. Here we're actually using specific amino acids and specific codons. So an important note that's going to come up later is that all amino acids start with methionine. So here's that amino acid, MET, that stands for methionine. It's specifically coded by the codon AUG. So it's very important to note that all proteins or polypeptides start with AUG, or methionine. Then we then have more codons, AAG, UUU, and then finally we have UAG, which is their stop codon. As the name implies, the stop codon tells the translation machinery to stop. It's very important to note here is that the stop codon does not code for an amino acid. So AUG, which codes for methionine and tells translation to start, does code for an amino acid, which is of course methionine. Here, the stop codon, though, UAG, does not code for an amino acid. So if I say you have 12 nucleotides with a start and stop codon, and I ask you how many amino acids does that code for, the answer is not 4 amino acids, it's 3 amino acids. So since there's a stop codon in there, that stop codon does not code for an amino acid, so it's actually 3 amino acids. So again, be aware that the stop codon does not code for an amino acid. So here's question 2, and this relates back to Mendelian genetics, specifically focusing on genotype and phenotype. So take some time to read this question and put in a preliminary answer on Blackboard. So here the answer is actually C. So as you can imagine, DNA contains our genes, which is of course why we call genotype. So it has the word genes in it. So DNA contains our genes and proteins contain our phenotype. So essentially the reason why my hair is black and why I have brown eyes, why I am as tall as I am, is all due to proteins. So proteins determine all those traits and more. So of these answers, C is the best. So DNA determines our genotype and proteins determine our phenotype. So we've talked a little bit about this already. So this is genetic code. So we talked about codons and how they specify certain amino acids. So the genetic code actually determines how we go from nucleotides to amino acids. The code is redundant, unambiguous, and universal. So actually almost all of life uses the same genetic code. So if I put an RNA into a yeast cell, so if I took a human RNA and put it into a yeast cell or a bacterial cell, they would actually be able to produce the same protein as a human. And that's because the genetic code is the same, so it's coding for the exact same protein there. So the same codons, or 3 nucleotide pieces, codes for the same exact amino acids as in bacteria, as in yeast, and as in humans. So to interpret the code, we actually use a codon table. So remember, a codon is each of those 3 nucleotide pieces of RNA. And so a codon table allows us to figure out what proteins are produced from certain sequences of RNA. So we talked about methionine, so this is our start codon. Methionine itself, though, is also an amino acid. And as you can see, methionine is coded by AUG. We also have our stop codons, which we talked about. So UAA, UAG, and UGA are the 3 stop codons. So if the translation machinery sees those 3 nucleotides, it's going to stop there and stop translation. And as you can see in the rest of the table, we have all other 3 nucleotide possibilities here. So for something like UUU, that codes for the amino acid PHE or phenylalanine. So really, again, the point of the codon table is allow us to go from RNA to protein. And as we said, it's unambiguous, redundant, and universal. So we can see the redundancy here. So for something like leucine, LEU, that's coded by 4 different codons. So again, it's redundant, so we have 4 codons coding for leucine. It's unambiguous, so as you can see, there's no ambiguity. So UCU is always going to code for serine, CCU is always going to code for proline, or PRO. So there's no ambiguity here. And as we discussed, it's also universal. So this codon table I can use for bacteria, I can use for yeast, I can use for humans, I can use for monkeys. For probably about 99.9% of life, you can use the codon table here. It's very rare that an organism uses a different codon table. For example, some viruses use separate codon tables. Also, I believe your mitochondria as well also uses a different codon table. But for the most part, in 99.9% of life, you can use this codon table right here. So the codon table, or genetic code itself, is universal. Okay, so now let's practice using the codon table. So imagine you have the RNA sequence below. Determine the amino acid sequence using the codon table on the right. So here the correct answer is C. So all you have to do is just read the codon table. So the first codon here is UUC. I've also been nice here and actually split these up into triplets for you already. So here we have triplet chunks already set out. So UUC, we just find what that is. So that is phenylalanine. So that is our first amino acid. Next we have CAU. It's histidine. We have GGU next, glycine or GLY, CGA, arginine, GGU, glycine, CGU, arginine. So that leaves us with C, C is the correct answer here. So for this question, I didn't actually have a start and stop codon. So it's important to remember that translation starts the start codon and stops the stop codon. So for this question, since there was no start or stop codon, I just want you to translate this directly as written. For exam questions though, I'll actually make sure to include a start and stop codon so that you know where translation starts and translation stops. I think we'll actually have some examples of that in future slides. So now we're going to go over the process of transcription in more detail. So remember that transcription is going from DNA to RNA, and that happens in the nucleus of the cell. So I'll first start off by showing you guys an animation of transcription.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So hopefully that gave you a pretty good idea of the process of transcription. Here's a nice figure from your textbook though that goes over the same exact thing that was shown in the animation. So there are really 3 steps of transcription. There's initiation, elongation, and termination. So initiation is the first step of transcription. So the first thing that happens is we have an enzyme come along called RNA polymerase. So RNA polymerase, as you can imagine, is very similar to DNA polymerase. So DNA polymerase was the enzyme that did DNA replication. Here, RNA polymerase binds to the promoter of DNA. So the promoter is actually a sequence of DNA that tells RNA polymerase to bind there. The RNA polymerase then binds to the promoter and pries apart the DNA. So in DNA replication, we heard of the enzyme helicase. So helicase unwinds the DNA. Here, RNA polymerase actually has helicase activity. So as opposed to having a separate enzyme do that, here RNA polymerase is actually able to separate those strands of DNA. So this is initiation of transcription. Next, we have elongation. So just like DNA polymerase, RNA polymerase can only add nucleotides to the 3' end of the strand. So here it would actually be a 3' end, and here would be the 5' end of our growing RNA strand. Also, as you can see, it's only copying one of the DNA strands. So in this case, the bottom DNA strand is our template strand. So again, as I said earlier, depending on the location of the promoter, we can have RNA polymerase heading right or left, or using the top or bottom strand. Here it's heading towards the right. So as you can see, the direction of transcription is towards the right, and it's using the bottom strand as the template. It can also head towards the left, and at the same time use the top strand as a template as well. So what determines the direction of RNA polymerase is the location of the promoter on the DNA strand. Here though, we're using the bottom strand as a template. So we just have RNA polymerase continue on and on. It's adding nucleotides to the 3' end of that growing RNA strand. Eventually though, it hits another sequence of DNA called the terminator sequence, or here the terminator DNA sequence. So this is a sequence of DNA that tells RNA polymerase to stop. So just like the promoter tells RNA polymerase to start, the terminator tells it to stop. Once that's done, RNA polymerase falls off and our RNA is completed. So again, these are the 3 steps of transcription: initiation, elongation, and termination. So one note I want to make here is that the promoter and terminator are different from the start and stop codon. So the promoter and terminator are all about transcription, the start and stop codon are all about translation. So the promoter determines the transcription start, and the terminator determines transcription end. The start codon determines translation start, and the stop codon determines translation stop. So please be aware of that. Don't confuse the promoter and terminator with the stop and start codon. So on this slide, I'm actually going to have you do an activity. So let's imagine you have the DNA sequence below. So here we have a double-stranded piece of DNA. And essentially what I want you to do is transcribe this DNA. So please pull out a piece of paper and do all the following steps. So show me transcription from initiation to termination, and make sure to include the promoter, the terminator, transcription bubble, RNA polymerase, directionality of both DNA and RNA, and the sequence of the new RNA. So essentially I want a little drawing that shows all these pieces in your answer. So here I've cheated a little bit, so I've gone a few steps ahead where we've actually separated the strands of DNA. So really quickly I'll draw out the first couple of steps that were skipped. So first we have double-stranded DNA. I have the 5' right here. So that means the 3' must be over here. On the bottom strand I have the 3' there, so the 5' must be there. We have the promoter on the bottom strand, right here. And the terminator must be somewhere far over here, likely at the end of the strand. So this is what it would look like before initiation. The next step, where we actually have initiation itself, RNA polymerase is going to come along and unwind these strands. And essentially that is what leads to what I have drawn below. So I'll clear out the slide so we have a little bit more room to write. So remember, RNA polymerase binds to the promoter. So it's going to bind this green box right here. And next we're going to have elongation. So essentially RNA polymerase is going to head towards the right and add nucleotides to the 3' end of the growing strand. So let's just add our directionality again. So here we have 3', here we have 5'. So remember, RNA polymerase is adding nucleotides to the 3' end of the growing strand. So let's start transcribing. So T pairs with A goes to U, C, G, U, A, G, C, U, A, and so on until we hit the terminator. So I actually haven't marked the terminator here, but it would be somewhere around here probably. So we mark this as the terminator. So again, this strand will just continue on until it hits the terminator. Once RNA polymerase hits that terminator, it falls off and transcription is done. So again, remember, RNA polymerase adds nucleotides to the 3' end of the growing strand. So since we have 3' here, this strand must be 5' here and 3' over here. So the growing strand itself is anti-parallel, and as you can see, RNA polymerase, if it was actually moving, would be adding nucleotides to the 3' end of that growing strand. So on the flip side of this, imagine if we had a promoter up here. So this was our promoter here instead of on the bottom strand. So if this is 5' here, this must be 3' here, and if we had RNA polymerase, it would actually be heading towards the left. And the way you know that is because the RNA strand is going to be anti-parallel. So if 3' is over here, 5' is over here, and 3' is over there. So be prepared to answer questions like this. I'll often ask you to transcribe a strand of DNA into RNA. So the major product of transcription is actually called messenger RNA. So messenger RNA specifically codes for proteins. As we're going to learn about in later chapters, there are actually different types of RNAs that don't specifically code for proteins, and those get different names. But here, mRNA, or messenger RNA, codes for proteins, and that's the major product of transcription. So looking at the whole cell, the majority of the cell's RNAs are messenger RNAs. So as you saw, DNA replication and transcription are very related. So in DNA replication, you have the replication bubble. In transcription, we have the transcription bubble. The main enzyme, as you know, in DNA replication is DNA polymerase. In transcription, we have RNA polymerase. Of course, the major product of DNA replication is DNA, and the major product of transcription is RNA. One major difference between DNA replication and transcription is the number of strands we produce. So in DNA replication, that's semi-conservative replication, so we produce 2 new strands, each paired with 1 old strand or parental strand. In transcription, we produce only 1 strand of RNA. In regards to the direction the enzymes go in, they both proceed in the 3' to 5' direction. So both DNA polymerase and RNA polymerase only add nucleotides to the 3' end of their growing strand. Also important to note is the fact that DNA replication relies on the enzyme helicase. So helicase unwinds the DNA, so it unravels it and melts it. For transcription, though, that activity is built into the RNA polymerase itself. So the RNA polymerase itself can unwind DNA. Here's question 4 for Blackboard, so true or false. As soon as transcription is finished, the mRNA can leave the nucleus. So if you've done the reading already, you should know the answer here is false. So before the RNA can leave the nucleus, it needs to be processed. So these are 3 or 4 major modifications that all RNAs have to go through before they can leave the nucleus. So as you can imagine, bacteria don't have this problem since they don't have a nucleus. So bacteria don't actually undergo RNA processing like eukaryotes do. So here are the major steps. So first we start off with a piece of DNA. Here we actually have a whole entire gene. You can imagine from here to here. This is the whole entire gene. And parts of that gene have exons and introns. So we're going to see exactly what those are in the next step when we actually look at the finished transcribed RNA. So that RNA itself also has exons and introns. Here, though, they actually are much more important. So introns actually stay inside the nucleus. So they actually get chopped out of the transcript, as you can see here. So they actually cut out. And the exons are then spliced together. So they're spliced into a single RNA transcript. So essentially we cut out the introns and we splice together the exons. We'll actually see this idea come back up in chapter 11 when we talk about gene regulation. But for now, know that introns are removed and exons are spliced together in the final transcript. Also be aware there's a cap added to the 5' end of the transcript and a tail added to the 3' end. So the cap is sometimes known as a methyl G or methyl guanine cap. So essentially it's a slightly modified G nucleotide. The tail is sometimes called a poly-A tail. So it's a polyadenosine tail, so a bunch of A's string together. So we have a methyl G 5' cap and a poly-A tail. So the cap and tail are both important for RNA stability. So once the RNA leaves the nucleus, it's going to be degraded very quickly if it didn't have that cap and tail. So together, exon splicing, the cap, and the tail all will allow the mRNA transcript to leave the nucleus. So as you can see, it leaves the nucleus, enters the cytoplasm of the cell, where it can then be translated into protein. So next we're going to talk about translation. So remember, this is going from mRNA to proteins. So the major players here are tRNAs, which we're going to talk about next, and ribosomes. So the ribosome itself is made up of small and large subunits. The ribosome is also a very unique enzyme compared to the other ones we've studied. So enzymes found in processes like photosynthesis and cellular respiration, all those enzymes are made from proteins. Here the ribosome, though, is actually made from both proteins and RNA. So that makes it a very interesting enzyme since it actually has RNAs that catalyze reactions as well as proteins that catalyze reactions. So again, most of the enzymes we've studied so far are proteins, but here we have an example where we have a protein and RNA-based enzyme. So here's a slide showing the structure of a tRNA. So tRNAs, as alluded to, are RNAs. So we're transcribed just like messenger RNAs, but here they don't code for proteins, and we're actually going to see what their function is as we walk through this slide. So tRNAs are actually kind of interesting because they form a 3D structure very similar to how a protein forms a 3D structure. Here you can see hydrogen bonds between bases. So we saw with messenger RNA, that's a single-stranded piece of RNA. Here, though, in this tRNA, we actually have 3D structure. So we actually have hydrogen bonding, making this into the shape we see right here. So it's sort of this L-shaped configuration. So really, tRNAs do the function of translating. So they take that RNA message and convert that into amino acids. And so they need two major parts to do that. They need the anticodon and the amino acid attachment site. So the anticodon right here binds to the RNA or messenger RNA. So these tRNAs understand the language of the messenger RNA. Attached to each tRNA is also an amino acid. So you can imagine if this was an AUG codon, it would have a methionine attached to it. So in that instance, the anticodon would recognize AUG, and this tRNA itself would have methionine already attached to it. So that's how we translate RNA into amino acids, is through these tRNAs. And that's again because I have an anticodon that recognizes RNA, and they have an amino acid that is attached to themselves. And so really these tRNAs are the basis for the codon table we have. So the tRNA that recognizes AUG has a methionine attached to it. Another example of this is the tRNA that recognizes ACG, so that has a threonine attached to it. So again, tRNAs is how we go from RNA to amino acids. So imagine we had a codon that has a sequence UCU. So what would you think the sequence of the anticodon would be? So here we'd have proper base pairing just as usual. So for UCU, we would have A, G, A in the anticodon. So normal base pairing is happening here. You can also even add directionality to this. So this would be the 5' here, 3' here, and this would be the 3' here, and 5' here. So same exact idea, we have proper base pairing and antiparallel strands between the codon and the anticodon.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anti-codon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
So here's a version of that animation that comes from your textbook. So step one is, of course, we have transcription. So we have RNA polymerase come along and generate the mRNA we're going to use for translation. So step two is something we didn't really talk about and is something that wasn't addressed in the video, but here's the charging of tRNAs. So charging is a technical term for the addition of amino acid to the tRNA. So as you can imagine, we just have a tRNA by itself. There's an amino acid, and an enzyme plus energy in the form of ATP comes along and binds that amino acid to the tRNA. So that's the process of tRNA charging. So step three is really where translation starts. So as you can see, the small subunit, the initiator tRNA, and large subunit bind to the strand. We then have movement of tRNAs and movement of the mRNA itself, as well as a growing polypeptide strand. Finally, the ribosome reaches the stop codon, so we have something bind the A site right here, and translation is terminated. So here's a little bit more of a zoomed in picture, so same exact idea. Here we actually have a 3D reconstruction of what the ribosome looks like. So it's actually very similar to what the textbook authors have drawn. So there's a large subunit right here, and a small subunit down here. We have our RNA strand, and you can see the tRNAs right there, so you have the characteristic L shape. Same exact idea on the right hand. We have the small and large subunit, and as you can see, we have 3 sites. The A site, the P site, and it isn't marked here, but we also have the E site. So for the most part, tRNAs go from A to E to E. So they progress in the leftward direction. The one exception of this is methionine. So methionine is our start codon. So the start codon actually starts in the P site, not in the A site. However, every other amino acid that comes through goes through A, P, and then E. And in the bottom right hand of the slide, we just have an example of what the ribosome looks like as it's translating some mRNA. So here's another activity. So this is going to ask you to draw translation instead of transcription this time. So I've given you another sequence from 5' to 3', and I want you to translate the sequence as well as draw out the major steps. So I think this is a very helpful activity for studying for exams, since it really has you think about the process as well as the steps that take place. So let's just draw a quick schematic of what this is going to look like. Let's go the 5' there, 3' there for your mRNA. And so what actually happens is we have the small ribosomal subunit start scanning across our messenger RNA. So it goes from 5' to 3', and it's looking for our start codon. So remember, translation starts with the start codon. So it's going to be looking for AUG. In that case, that's right here. So that is our start codon. So it's very important to remember that translation starts at the start codon and ends at the stop codon. So in this case, all the stuff before AUG is useless. So don't worry about that. That's before translation starts. It's still part of the mRNA, but it's not part of the amino acid. So it's not going to be translated. So again, we have AUG. That's where the small ribosomal subunit is going to bind. Next, we have a tRNA. In this case, methionine. That's going to come along and bind. Remember, the tRNA anticodon is complementary to the codon of the mRNA. So in this case, it's going to be U, A, C. And then finally, we add our large subunit, which has the A site, and the E site, and the P in the middle, which I can't really add to this figure right here. So all this ribosome is going to do in the next step is just elongate the amino acid or polypeptide strand and just keep on adding amino acids until it hits the stop codon. So really just to translate this message right here, we just split it into chunks of 3. So AUG is one chunk, AU is a second chunk, UG is a second chunk, AGA, then UAA. So AUU would code for isoleucine, UGG codes for tryptophan, AGA codes for arginine, and UAA is for stop codon. So remember, translation starts at the start codon, ends at the stop codon. So just like everything before the start codon was junk, here everything after the stop codon is junk as well. So ignore that, stop translation at the stop codon. So as you saw in the animation, a release factor will come along, bind to the A site. So this is a release factor. And that tells the ribosome its job is done and it's time to stop translating. So hopefully your drawing is a little bit neater than mine, but I hope this gave you a good idea of what translation looks like and also the answer you should have generated for this question. So again, just like transcription questions where I ask you to transcribe DNA, I'll also be asking you to translate mRNA. So to finish this lecture, I want to talk about DNA mutations. So here we have 4 different types of DNA mutations. So here we have the 3 major types of DNA mutations. So substitutions, deletions, and insertions. So I think these are all fairly intuitive to understand. So at the very top strand, we actually have our normal strand. So it codes for methionine, lysine, phenylalanine, glycine, and alanine. In a substitution, we have a change in the nucleotide. So we go from G to A. In deletions, we have a nucleotide deleted. Here we have a U deleted. And then finally in insertions, we have a nucleotide inserted. One thing that's misleading about this slide is it looks like there's mutations in the messenger RNA. So often mRNA is not mutated itself. So that's usually at the DNA level. So there would be some sort of substitution, insertion, or deletion in the DNA level that changes the mRNA that's made. So that's what's misleading about this slide, is that it's showing mRNA changing when really it should be the DNA strands themselves. So as you saw, insertions, deletions, substitutions can result in amino acid changes. So depending on the change that happens to the amino acid, that gets a name. So it's important to note that these names are independent of what happens to DNA. So the last slide was focusing at the level of DNA. Here we're looking at the protein level. So what's happening to the amino acids? So the first type of mutation is a silent mutation. So this is when the amino acid sequence does not change. There's still a nucleotide change though. So here we're changing an A to a T. As we saw, the codon table is redundant. So in this case, even though there was a mutation, both codons still coded for valine or VAL. The next type of mutation is a missense mutation. So just like we have a nucleotide substitution, here we have an amino acid substitution. So we went from a proline, or PRO, to a threonine. Next, we have a nonsense mutation. So this is when you have an early stop codon. So that amino acid was supposed to be tyrosine, but here it changed to a stop codon. And lastly, we have frame shift mutations. So a frame shift mutation is actually when the ribosome gets knocked off by 1 or 2 nucleotides.